every single time that, a sprinter explodes off the starting blocks or a powerlifter hoists just a massive amount of weight, they're relying on a biological machine that is actually smaller than a single bacterium.
Right, which is wild to think about.
It really is. I mean, we so easily picture human strength as this large macro-level force, right? Like bulging biceps, sweeping quads, just explosive power. But all of that movement is actually generated by a microscopic engine that's roughly 2 micrometers long.
Yeah, it completely flips your perspective on how the human body operates. We are talking about a structure so unbelievably small that, well, thousands of them can stack end to end inside a single muscle cell, and yet this tiny engine completely dictates our physical reality.
And that is exactly our mission for today's deep dive. We are unpacking a really fascinating stack of anatomy and physiology sources to explore the ultimate contractile unit of striated muscle, the sarcomere.
Sarcomere, yes.
We are going to look at the machinery responsible for everything from that sprinter speed to the biological realities of aging, space travel, and even forensic science. So it's a huge topic for you today.
It's a profound topic because we are not just looking at dry anatomical definitions here. We are exploring how muscles actually generate force at a molecular level. And perhaps more importantly, I think the story of the sarcomere is a perfect case study in how deeply we can misunderstand our own biology.
Oh, absolutely.
Yeah, and how scientific observation eventually, corrects the record over time.
Well, the history here is just wild because to truly grasp how the sarcomere works, we really have to look at how wrong the initial theories were. Like, scientists have known about the stripes in muscle tissue for well over a century, right?
Yeah, early microscopes could clearly see a pattern.
Right, so when 19th century microscopists looked at muscle flesh, they saw this distinct, almost barcode-like pattern, alternating dark and light bands.
Exactly. And they eventually named these repeating units sarcomeres. They pulled that from the Greek words sarx, which means flesh, and meros for part.
Flesh part.
Flesh part, exactly. So they mapped the visual boundaries really early on. But looking at a machine doesn't actually tell you how it runs. The mechanism of contraction was a complete mystery.
Right, because they could see the muscle shortening overall, and they could see the bands under the microscope, so they just assumed the material itself was shrinking.
That was the dominant assumption for decades. Up until 1954, the prevailing idea in the scientific community was the folding filament theory.
The folding filament theory, okay.
Yeah. The thought was that muscle tissue contained these long, continuous proteins that simply, folded up on themselves to get shorter. It made intuitive sense based on the macroscopic world.
Like an accordion or something.
Yeah, exactly. If you want a string or a spring to get shorter, you coil it or you compress it like an accordion.
But then, in 1954, that accordion theory gets completely shattered by what the sources describe as just one of those staggering historical coincidences in modern biology.
Oh, it really is. It's crazy. On May 22, 1954, the journal Nature published 2 back-to-back papers that completely revolutionized muscle physiology. One paper was authored by Andrew F. Huxley and Rolf Niedergerk, who were bouncing light through living frog muscle fibers to measure the bands.
Okay.
And the other was by Hugh E. Huxley and Gene Hansen, who were using electron microscopes and X-rays to look at the isolated proteins.
And the craziest part about this, and I had to read this twice in the sources, is that Andrew Huxley and Hugh Huxley were totally unrelated.
No relation at all.
They were working entirely independently in totally different labs, using different techniques. They just happened to share a last name, and they just happened to discover the exact same radical biological truth at the exact same time.
It's a true moment of convergent scientific discovery. Both teams realized that the folding filament model was just physically impossible based on what they were seeing. Because they noticed that when a muscle contracted, the dark bands in that barcode pattern, they didn't shrink or fold at all. They stayed the exact same width. So to explain this, they proposed the sliding filament theory.
Okay, so instead of a single spring folding up, muscle shortens because distinct pieces are physically sliding past each other, like having 2 interleaved combs being pushed together.
Yes.
The plastic teeth of the combs don't shrink or coil, they just overlap more deeply.
That analogy captures the mechanics perfectly. The physical filaments inside the sarcomere do not change length, They slide past one another, basically telescoping the entire unit shorter without any individual piece actually shrinking.
Wow.
Yeah, and that single revelation falsified the accordion model forever. It became the foundational core of modern muscle physiology.
Well, if the components are sliding rather than shrinking, we should probably map out the physical geography of this microscopic space for you listening. Like, what exactly are these cones made of?
Let's visualize the architecture. The boundaries of a single sarcomere run from one structural wall to the next, and we call these walls Z-discs.
Z-discs.
Right. They are essentially protein lattices that cap the ends of our 2-micrometer unit. Anchored to these Z-disc walls, projecting inward toward the center, are the thin filaments. And these are made primarily of a protein called actin.
Okay, so the actin acts like thin ropes tethered to the outer walls.
Exactly. Thin ropes. Then, floating in the very center of the sarcomere, overlapping with those thin ropes, are the thick filaments. These are made of a protein called myosin.
Actin and myosin.
Yep. And this arrangement is what creates that visual barcode that early scientists saw. The areas where you only have thin actin filaments, they let a lot of light through. That's the light band.
Makes sense.
But the areas where the thick myosin cables sit, those block the light, creating the dark band.
You know, this might actually be a good time to clear up a structural misconception that constantly pops up in anatomy discussions.
Right.
When we talk about filaments and fibers in these tiny units, it is incredibly easy to lose a sense of scale. A sarcomere is not a muscle cell, right?
No, it is vital to separate those concepts. Think of a muscle like a Russian nesting doll.
Okay.
The muscle itself is made of bundles of fibers. A muscle fiber is the actual biological cell, and it is massive for a cell, sometimes stretching several centimeters long, packed with multiple nuclei.
Wow, a single cell is that long.
Yes. And inside that giant fiber cell are long rod-like tubes called myofibrils. And inside those myofibrils, stacked end-to-end like thousands of train cars, are the sarcomeres.
So the sarcomere is just one tiny segment of the train track inside the larger cell.
Yes. When your bicep curls, it isn't one sarcomere doing the work. It is tens of thousands of these tiny units in series, all microscopically sliding their filaments closer together at the exact same moment. That creates the macroscopic movement.
Okay, I have to bring this up because there's another protein in this structure mentioned in the sources, sitting alongside the actin and myosin called titin. And its sheer scale is just fascinating to me.
Titin is an absolute marvel. It functions as a massive, passive elastic spring.
A spring, okay.
Yeah, it basically tethers the thick myosin filaments to the Z-disc walls. Its primary job is to provide structural integrity and prevent the sarcomere from being pulled completely apart when a muscle is severely stretched.
The sources note it actually holds a Guinness World Record for being the largest known protein in the human body, which is crazy.
The size is staggering. To put it in perspective for you, if an average cellular protein were the size of a toy car, titin would be a massive freight train.
A freight train.
A single molecule can be composed of over 33,000 amino acids.
That is absurd.
It is. And as the muscle stretches, specific folded domains of the titin molecule literally unfold one by one, providing an escalating elastic resistance. And much like the sliding filament theory, titin had a dual discovery.
Oh, really? Another one.
Yeah, in the late 1970s, researchers in Japan found this massive spring and called it connectin, while a completely independent lab in the US found it a year later and named it titin. It took the scientific community some time to realize they were cataloging the exact same molecular bungee cord.
So we have our structural walls, our titin springs, our thin actin ropes, and our thick myosin cables in the center. But these interleaved cones don't just push themselves together magically. What physically drives the sliding motion?
The movement is driven by what biophysicists call the cross-bridge cycle. It is a continuous asynchronous mechanical motor, and it's fueled by ATP, adenosine triphosphate, which is the primary energy currency of your cells.
Okay, but how does the ATP physically make the filaments move?
It really comes down to the shape of the myosin. Those thick central cables aren't just smooth rods. They are covered in thousands of tiny globular myosin heads that project outward, kind of resembling little golf clubs.
The little golf clubs, okay.
And these heads have a strong chemical affinity for the thin actin filaments. They basically want to grab on.
Walk us through the cycle then. How do the little golf clubs actually pull the actin ropes?
It is a 5-step mechanical loop. First, the myosin head starts out already attached to the actin. A molecule of ATP binds to the head, which actually causes it to release its grip.
Wait, it releases?
Yeah, it lets go. Second, the myosin head hydrolyzes the ATP. It breaks a chemical bond, turning ATP into ADP and a loose phosphate. Breaking that bond releases energy, which physically pivots the myosin head backward into a cocked high-energy position, much like pulling back the hammer on a gun.
So the energy from the ATP doesn't cause the pull itself, it just cocks the spring.
Precisely. In step 3, that newly cocked head binds to a binding site further down the actin filament, forming what we call a cross bridge. Then step 4 is the actual power stroke. The loose phosphate falls away, and the myosin head violently snaps back to its original resting position, dragging the actin filament roughly 10 nanometers toward the center of the sarcomere.
10 nanometers. That is tiny.
Very tiny. And finally, in step 5, the ADP falls away, leaving the head empty but still gripping the actin, just waiting for the next ATT molecule to arrive and restart the cycle.
And because there are thousands of these little heads grabbing, pulling, and releasing at different times, the overall pull is smooth rather than jerky.
Exactly.
But this raises a major point of confusion for me, and I'm sure for you listening too, because when it comes to muscle function, cramps, or athletic performance, the mainstream conversation always revolves around calcium. But we just went through the entire motorcycle, and calcium wasn't involved at all. You just said ATP is doing all the work.
I know, it is one of the most persistent misunderstandings in biology. People assume calcium is the metabolic fuel for muscle contraction. It is absolutely not.
Wait, so if ATP is the fuel actually doing the work, what on earth is calcium doing? Is calcium just like the bouncer moving a velvet rope so the ATP can actually get to the bar?
I love that. Yes, that is a highly accurate way to visualize it. The velvet rope in this scenario is a long regulatory protein called tropomyosin. When a muscle is relaxed, tropomyosin physically coils around the thin actin filament and covers up all of those binding sites. So even if the myosin heads are fully cocked with ATP and totally ready to pull, they just can't grab the actin. The velvet rope is in the way.
So how does the bouncer know when to move the rope?
The bouncer is a secondary protein complex called troponin, which sits right on top of the tropomyosin rope. When your brain decides to move an arm or take a step, an electrical nerve impulse travels down into the muscle fiber. This electrical signal triggers an internal storage network inside the cell, the sarcoplasmic reticulum, to suddenly flood the space around the sarcomeres with calcium ions.
And the calcium interacts with the bouncer.
Yes. Calcium binds directly to troponin. When that chemical binding occurs, troponin physically changes shape. That shape change yanks the tropomyosin rope aside, exposing all the binding sites. The cocked myosin heads immediately attach, the power strokes occur, and the muscle contracts. This entire sequence is known as excitation-contraction coupling.
That is brilliant.
And the moment the electrical signal stops, cellular pumps quickly vacuum the calcium back into storage, the velvet rope slides back over the actin, and the contraction ends. Calcium is the biological switch. ATP is the mechanical fuel.
That makes so much sense.
Yeah.
Okay, if calcium flips the switch and ATP pays for the movement, does that mean simply maximizing the sliding equals maximum strength? Like if I overlap those combs as far as they can possibly go, do I generate the most force?
Actually, no. And proving that required a landmark experiment in 1966 that defined the physical limitations of the sarcomere. 3 scientists, Gordon, Huxley, and Julian, designed this remarkable study using single microscopic muscle fibers isolated from frogs.
Single fibers. That sounds incredibly difficult.
It was incredibly delicate work. They built an apparatus that could precisely stretch or compress individual sarcomeres to exact lengths, stimulate them to contract, and measure the precise amount of force they produced.
So they were looking for the sweet spot, basically, the Goldilocks zone of force generation.
And they found it. They discovered that if you stretch a sarcomere way out past 3.6 micrometers, the muscle generates 0 active force. At that extreme length, the thin actin and thick myosin are pulled entirely past one another. There is no overlap.
So the velvet rope is down, the heads are cocked, but there is literally nothing for the myosin to grab onto.
Exactly. It's like reaching out for a handshake and missing entirely. And on the opposite end of the spectrum, if you compress it too much before stimulating it, it also fails.
Because it just crashes into itself.
Yeah, when the sarcomere is severely shortened before a contraction even begins, the thin actin filaments slide so far toward the center that they crash into one another. The thick myosin cables ram straight into the structural Z-disc walls. The whole physical architecture buckles and force plummets.
So what was the optimal length? Where did the force actually peak?
The empirical data showed a peak resting length of roughly 2.0 to 2.2 micrometers. At that precise length, you have the maximum mathematical number of myosin heads perfectly aligned with exposed actin binding sites, with plenty of physical room left to actually slide.
That is perfectly balanced.
And what made this 1966 experiment so monumental wasn't just finding a number. It was the ultimate proof of the sliding filament theory. They proved that a muscle strength depends directly on the surface area of overlapping filaments, not on the filament's changing length.
So we have this elegantly optimized 2-micrometer machine. But to really appreciate its importance, I think we need to scale up. Why does understanding this tiny structure matter for our broader health, for our survival?
Because every macro change in your muscular system is dictated by the biology of the sarcomere. When you lift heavy weights at the gym, the extreme mechanical tension literally pulls on the Z-disc walls.
Oh, wow.
Yeah, and this physical stress triggers a process called mechanotransduction. The physical deformation sends chemical signals to the cell's nucleus, telling the body to synthesize new actin and myosin proteins.
So the cell builds more engines to handle the stress.
Exactly. Traditional heavy lifting causes the body to add new sarcomeres in parallel, so side by side, which physically thickens the muscle fiber. That is the fundamental mechanism behind hypertrophy, or gaining muscle mass. Other movements, like loaded stretches, can tell the body to add sarcomeres in series, physically lengthening the myofibrils.
But biological efficiency works both ways, right? If the body doesn't sense that mechanical stress, it doesn't want to pay the metabolic cost to keep all those engines running.
Maintaining billions of sarcomeres requires a massive amount of ATP. The body is relentlessly efficient. We see this vividly in spaceflight, actually.
Right, the sources mentioned astronauts.
Yeah, when astronauts live on the International Space Station, the microgravity environment removes the constant mechanical load of Earth's gravity. Without that physical tension pulling on the Z-discs every time they stand up, the cells just stop synthesizing new contractile proteins.
And it gets worse than that, right?
It does. Cellular recycling mechanisms actively dismantle the unused sarcomeres to save energy.
Which perfectly explains why astronauts have to endure hours of just intense resistance training every single day on the ISS just to mitigate the loss.
Exactly. And the same fundamental mechanism applies to aging. Sarcopenia, which is the age-related loss of muscle mass, is essentially the progressive loss of functional sarcomeres, compounded by changes in the nervous system's ability to activate them. It leads directly to frailty and loss of independence as we age.
And our sources also point out that the sarcomere plays a critical role in some very sudden, severe heart conditions.
Yes, it governs cardiac life and death. Your heart is composed of specialized striated muscle. If a person inherits a genetic mutation in the genes responsible for building sarcomeric proteins, specifically the genes that code for cardiac myosin or regulatory proteins, the individual engines are structurally weak.
So how does a weak microscopic engine lead to a thickened, enlarged heart? Because that's what we usually hear about with these conditions.
It is a compensatory mechanism. Because the individual sarcomeres are weak, the heart struggles to maintain adequate blood pressure. The cardiac cells essentially panic.
Oh, no.
They attempt to solve the problem by frantically synthesizing millions of additional sarcomeres. This overbuilding causes the walls of the heart to thicken abnormally, a condition called hypertrophic cardiomyopathy. The electrical signaling gets completely disrupted through this dense, chaotic tissue, making it a leading cause of sudden cardiac arrest in young, otherwise completely healthy athletes.
That is terrifying,
Yeah.
but it makes so much sense biologically. Speaking of life and death, there is a connection in the notes regarding the ATP cycle that explains something fundamental about human mortality. And this kind of blew my mind.
You're talking about the mechanism behind rigor mortis.
Right. So we established earlier that living muscle requires ATP to bind to the myosin head so it can detach from the actin. The energy is quite literally required to let go.
That is the key biological reality. Living muscle needs ATP to break the cross bridges just as much as it needs ATP to prime the next stroke.
So if you follow the logic, when a person dies, cellular respiration ceases. They stop producing ATP. That means they don't have the energy fuel to contract. But crucially, they lack the chemical key to detach.
Yes.
The myosin heads are just permanently locked onto the actin.
The engine jams. Rigor mortis is a passive locking, a molecular rigor, not an active contraction. Whatever myosin heads happen to be attached the moment ATP depletion reached a critical threshold, they simply cannot let go. The entire muscular system locks into a rigid framework.
But how does that timeline inform forensic science? Because rigor mortis obviously doesn't last forever. A body doesn't stay stiff permanently.
Forensic pathologists use the predictability of this biological failure In a body at normal room temperature, rigor mortis typically begins setting in roughly 2 to 6 hours post-mortem as the residual ATP finally runs out.
Right.
The stiffness peaks around 12 hours, but you're right, it doesn't last forever. But the locks never actually unjam. There is no new ATP to release them.
So what happens?
Over the following 24 to 48 hours, the cellular environment breaks down completely. Enzymes called proteases, which normally act as controlled cellular recyclers, go rogue. They physically degrade the actin and myosin proteins.
Oh, so the door doesn't unlock, the door just gets kicked down and chewed up.
Exactly. The tissue itself degrades and the stiffness naturally resolves. It is a pretty grim biological timeline, but it perfectly illustrates how entirely dependent our macroexistence is on this microscopic cycle.
It is just a remarkable journey of discovery for you listening. I mean, here we started with early scientists looking through rudimentary microscopes, trying to guess why muscle tissue looked like a barcode. We saw how a massive coincidence in 1954 finally proved that the muscle isn't folding like an accordion, but sliding like interleaved combs.
Right.
And then we explored how calcium acts as the cellular switch to expose the binding sites, while ATP provides the mechanical energy for the golf club heads to pull. We unpacked how mechanical tension commands the body to build more of these 2-micrometer engines, and how the complete loss of ATP results in the ultimate biological lockup.
It is a very dense amount of physiology, but it is so important to realize that the textbook on the sarcomere is not closed.
Oh, really?
The science of how this motor operates is continually refined. For example, Hugh Huxley actually proposed the idea that the myosin head acts like a swinging lever arm way back in 1969. But it wasn't until 1993, 24 years later, that biophysicists managed to map the exact crystal structure of the protein and visually confirm that the lever hypothesis was physically possible.
And the sources mention an ongoing debate today regarding something called loose coupling. What exactly are biophysicists arguing about now?
Well, the classic textbook model assumes a perfect 1-to-1 ratio. One molecule of ATP hydrolyzes, the myosin head does one clean 10-nanometer pivot, and then it waits.
Sounds efficient.
It does. But modern biophysicists use highly advanced tools called optical traps. They use precisely focused lasers to hold a single actin filament and a single myosin head in suspension to measure the microscopic forces.
Wait, they are measuring the physical pull of a single protein with lasers?
Yes, it's incredible technology. And their data suggests the cycle might not be a rigid 1-to-1 lockstep. Loose coupling asks whether one ATP molecule might actually allow the myosin head to take multiple erratic, tiny substeps along the actin before fully detaching.
Oh.
Yeah, is the motor a perfect discrete machine, or is it a slightly chaotic, loosely coupled engine? We don't entirely know yet.
Which leaves us with a totally fascinating thought for you to mull over. Right now, as you are going about your day, taking a breath, shifting your posture, or walking down the street, there are millions of these microscopic ATP-fueled engines operating inside you in asynchronous harmony.
Absolutely flawless harmony.
It happens flawlessly, millions of times a second. But if a mechanism this vital to human movement was fundamentally misunderstood by the smartest people on Earth until the 1950s, what other perfect biological mysteries are operating inside you right now, just waiting for the next massive scientific shift to finally explain them?